For the final section in this chapter, we're going to be focusing more on the cell membrane itself, because it is actually incredibly important to the cell and the cell function. So cell membranes have two major functions. First is kind of the obvious one. They form a boundary between inside and outside of the cell. So they protect the cell from a lot of different things in its environment, and also controls, how much goes in and out of the cell. Okay, and then their second, of course, is the control of the passage of materials into and out of the cell. Okay, so when we look at the cell membrane, generally we think of it as just a sheet kind of surrounding the cell itself. Just a wall that, it's not a whole lot of interest in it. But the cell membrane actually has a lot going on inside of it. So you have the cell membrane itself made up of phospholipids, and they form what's known as a phospholipid bilayer. So it means that the membrane actually has two rows of phospholipids. Okay? So if you remember about the phospholipids, they have charged head groups. Okay? So these pink parts are negatively charged. Okay? And that's true on the top and the bottom layer, okay? And then the part inside is actually nonpolar. Okay, so we'll just label that as NP. So it has a very different chemical structure, basically, whenever you're referring to the top or bottom of it versus the inside of the membrane. Then on top of that, you also have different molecules inside, contained inside the membrane itself. Okay, so you've got these little green ones here that are designed to help kind of keep the membrane fluid. You also have your proteins here and here. And then you have carbohydrates, okay, which are on the outside of the cell for signaling. Okay. So the cell membrane, again, is made up of your phospholipid bilayer. Okay? You also have other molecules embedded in the membrane. The proteins that I was talking about, protein channels, then the carbohydrate chains and your cholesterols. Okay, so there's a lot going on in this membrane that you don't necessarily see when you just look at an image of the cell itself. Now, this model of the membrane is known as the fluid mosaic model. So the fluid mosaic model describes the arrangement of molecules inside the membrane. And it's broken down into two parts, really. Fluid, because the membrane is flexible. All of the components inside the membrane can shift and slide, including the phospholipids. So the phospholipids can actually flip from the inside of the cell to the outside of the cell and back. Okay? It all depends on, what the cell needs from its membrane. And then it's also referred to as mosaic because there are a variety of molecules that stud both the inside and outside of the cell. So if you were to look at it from above, okay, if you're just looking at the membrane, the cell membrane from above, everything is kind of moving around. So while the membrane itself is relatively solid, okay, it's still moving around more like a fluid. And then the mosaic part of it comes in because if you've ever looked at a mosaic, it uses little tiny pieces of different colors in order to make up an entire image. And this is kind of the same way that the plasma membrane, the cell membrane, works. You've got all of these little pieces everywhere. You've got phospholipids, you've got the cholesterols, you've got carbohydrates and different proteins, and they're all spaced around to kind of make up this mosaic. Now the cell membrane is also known as something that is selectively permeable. So permeable means that it can be penetrated. You can have things go across the membrane. Selectively means that the membrane basically gets to choose. So some molecules can pass through the membrane, can cross it. Others can't. So if you look at this image here, you've got the small blue spheres. Well, they either have the correct chemical composition or the correct size, or both, that they can cross through the membrane. But the orange ones here are probably too large. They might be negatively charged, so they won't contact the phospholipids, which also have that negative charge. So they can't cross the membrane. Okay? So the blue ones can go across, the orange ones can't. Now, when you're talking about the membrane, okay, some of the proteins that are studded into the membrane are what's known as receptors. So receptors are kind of how the cells talk to each other. Okay, so receptors bind with ligands. Some people call them ligands. I've always known it as ligands. But those are specific molecules that bind the receptor. Okay, the receptors themselves, once they bind these ligands, can change shape. And that can create kind of a signal, what's called a signaling cascade. Okay, where the receptor changes shape, and then that causes a change in a different molecule, and that causes a change in a different, so on and so forth, until a response has been activated. Okay, so it's a relatively complex, like step-by-step process, and a lot of research is actually going into each individual type of receptor and the signaling cascade that they can trigger. So there are two major types of receptors. The first is what's known as an intracellular. So if you think about intra and cellular, it means that it has to cross inside the cell. So the ligand can be found outside the cell, but in order for it to bind to the receptor, it has to cross to the inside of the cell. So these ligands can cross the cell membrane. Okay, the membrane, the selectively permeable membrane, decides that these ligands can come into the cell. And once those ligands are into the cell, okay, they bind the receptor that they can find inside the cell. And then that creates this response. Okay, so intracellular means that the ligand itself has to cross into the cell. Your second type is known as membrane receptors. Okay, so this tells you that the receptors are found inside the membrane. Okay, so they're one of those proteins that studs the membrane. Okay, and these will also change shape once they bind a ligand. Okay, but remember, the ligand in this case doesn't have to cross that membrane. It can be, well, it is bound outside of the cell. Okay, so this could have, different advantages depending on what the molecule is that is the ligand, depending on, concentration outside of the cell versus inside the cell, all different kinds of things, okay? But as you can see, in this case, we have, let me get back to here we go. In this case, we have our membrane-bound receptor here, okay, and it is inactive because it doesn't have a ligand bound to it. But then you see this one here, it binds the ligand, which changes the shape of the receptor, and then it can bind something from inside the cell. Okay, now that's one type of membrane receptor. Another type is kind of the opposite. It's already bound to something that's inside the cell, and when the ligand binds, it releases that secondary molecule. Okay, both are ways of starting another signaling cascade. Okay. So, when we talk about intracellular receptors, okay, have to talk about the ligand getting into the cell. Now, there are three major ways that a ligand can get into the cell. Okay, first up, we've got passive transport. So, if you talk about passive, okay, it's not expending any energy. So, passive transport doesn't require energy to move across the membrane. It uses what's known as a concentration gradient. So we'll go into that in just a second. There are two types of passive transport. There is what's known as diffusion, and there's osmosis. So osmosis deals specifically with passage of water through the cell membrane. Okay, so osmosis is a type of diffusion, but not all diffusion is osmosis. It's one of those weird things. Okay, so again, osmosis only deals with water crossing the membrane. Diffusion deals with anything passively crossing the membrane. So molecules diffuse down the concentration gradient. Okay, so they go from areas of high concentration, which is generally outside the cell, to areas of low concentration. So diffusion involves any molecule, right? So you see you've got your high concentration on the left here, and then it will pass through the membrane. So the membrane would be right about here, okay? And then you have the lower concentration inside the cell. So diffusion passively moves any kind of molecule down the concentration gradient. Osmosis, again, is the diffusion of water molecules, but it still works in the same way. Okay, it's still going to go from high concentration to low concentration. So that is one of the things that inside the cell, it has to be very careful about maintaining. So if you have a nutrient that is incredibly important to the function of the cell, but it has a high concentration inside the cell, it's not going to be getting any more of that coming in. Okay? So the cell is actually designed to, as soon as that molecule diffuses into the cell, it binds it with something else, and then it keeps its concentration inside the cell low. So it's always a net influx of these nutrients. Okay? Diffusion and osmosis are always working to move things into the cell. Okay? It's not always, but generally the cell tries to keep all of these nutrients at a very low concentration inside of the cell. And that's because, it's using those molecules as they're coming in and keeping that concentration low. So, solutions with diffusion. So this is when we're talking about movement across a selectively permeable membrane. Okay, and this is also talking about cells placed in these kind of solutions. So you have the three different types of solutions. You have what's known as isotonic. So an isotonic solution has concentrations of a particular nutrient inside and outside of the cell equal. So they are the same. Now this is kind of, if you're talking about water specifically, this is a good state for the cell to be in. Okay, so concentrations inside and outside of the cell are the same. You don't have a concentration gradient. So you're not going to have a net influx of water. You're not going to have a net outflux of water. Now when you go up to what's known as a hypertonic, okay, you have a concentration higher outside of the cell. So the cell is always trying to reach a balance, an equilibrium between those concentrations. So if the concentration is higher outside of the cell, you're going to have a net flow of water out of the cell. Now, this may not seem like it makes a whole lot of sense, okay? But one thing to remember is that the concentration of whatever nutrient it's talking about here, that nutrient cannot go into the cell. Okay, so if we, excuse my drawing here because my mouse is being weird, but you're not going to have that nutrient flowing into the cell. So in order for the cell to try to equalize the concentration, it has to let water out of the cell. Okay, and so when it pushes that water out of the cell itself shrinks, okay, because water is a large part of the cytoplasm. So when it's getting pushed out, the cell shrinks, and eventually it can die from that, okay? And then you have the other way. You have hypotonic. So hypotonic has a concentration higher inside the cell, okay? And again, because that nutrient that we're talking about here can't cross the membrane, you're going to get a net flow of water into the cell. Now that might not sound all that bad, okay? But your cell is going to swell, and eventually it will burst. Okay, so hypertonic causes cell lysing is what it's called. It lyses the cell, so it bursts open. Okay, so we're going to pause here and get to the next video.